Today we'll be going over transcription and translation. We'll first begin by going over the central dogma of biology, in which we take DNA from the nucleus, we transcribe it into RNA or mRNA, which is then sent out of the nucleus, and it undergoes translation via a ribosome. All right, so beginning with transcription. Transcription occurs in the nucleus of the cell, like we mentioned before, and the created RNA is released in the cytoplasm. There are 3 steps to transcription, which are initiation, elongation, and termination, which we'll go over briefly here in a minute. Beginning with initiation, the first thing to understand is how the cell knows what part of the DNA needs to be used to create the new RNA. DNA molecules contain what is known as a promoter. This is a section of DNA that includes a sequence of TATA, or thymine, adenine, thymine, adenine. It then continues and is about 25 nucleotides from the starting point. The enzyme RNA polymerase II recognizes that promoter and binds to the DNA strand. Transcription factors then bind to that polymerase to create the transcription initiation complex. RNA polymerase II then unwinds the double helix of DNA and begins to synthesize RNA from that strand. Again, what I've just gone over is illustrated here in this diagram where we have the sequence of TATA followed by the rest of the nucleotide bases. And then we have the transcription factors that come in here. And then we have the complex form here at the bottom. All right, so once initiation has occurred, we have what is called elongation. And now that the RNA synthesis has started, the RNA polymerase continues down this DNA strand. While it continues down the strand, it will continue to unwind the DNA, but it will also rewind that part that is already passed over, which you can see here as it is moving forward. We are opening up the DNA strand, and as it passes through, we close it back up. RNA polymerase adds the RNA nucleotides to the strand, and an important note that is instead of adding thymine, RNA contains a nucleotide uracil in its place. We still have cysteine and guanine, but instead of adenine and thymine, we do have adenine and uracil. So we have A-U bonds, which you can see an example of right here. Where it used to be A and T, it is now a U that the A is associated with. So once elongation has occurred, we then terminate. And the way we do that is with the newly synthesized RNA strand, we cut it off of the DNA and prepare it for its role in the cell. The signal will cause RNA polymerase to break off from the DNA, and with it will go the RNA. The RNA strand must then be prepared for what is going to go to, which is the ribosome in the cytoplasm. First, it is modified with a nucleotide guanine and then is added to the 5' end of the strand, which is often called the 5' cap, which we can see right here. Right here with the 5' end. Then another enzyme adds anywhere from 50 to 250 nucleotides of adenine to the 3' end, which we call that poly-A tel. Which again here is a 3' end, poly-A tel. To create a protective tel, often known as the poly-A tel. Finally, the RNA is cut down to what is needed for the actual gene. RNA is built of introns and extrons, where extrons, as we see for one right here, exons are the part of the RNA cell that the cell actually needs, and is what's going to be expressed in the gene, which will be used to produce the protein. We also have these introns, and the introns can be cut out, leaving what needs to be expressed. And so that will leave the complete molecule of mRNA. So some of the translation components we have transcription was what we just did. Translation components will be using the mRNA that we just synthesized or just replicated off of the DNA strand, the tRNA, and the ribosome. What's important to mention here is that the mRNA was produced from the transcription of the DNA, and that the tRNA and the ribosome were already in the cytoplasm. So here we have on the right just kind of a very general diagram consisting of all the different parts we're about to talk about. So if you ever want to reference back to this diagram, please do. All right, first we're going to talk about the mRNA. which again was what we produced via transcription. And it is what is essentially the blueprints for the protein that we've taken from the DNA, and we are just sending to the ribosome so that the protein can be produced. Going on to tRNA. tRNA is a molecule like RNA. However, it is folded slightly different so that it can bind to an amino acid and also bind to mRNA. So for example, here at the top, we have the binding site where we can carry an amino acid. And if we look at the bottom, we have the anticodon, which is going to connect and be represented on the mRNA strand. Once these match, we're then able to pull it into the ribosome and continue on with translation. So the ribosome. The ribosome is a protein or an enzyme consisting of two subunits, the small subunit, which we see on the bottom here, and the large subunit on the top. It is good to note that the small subunit is what we will interact with first, and the large subunit will come in later on. Also, the large subunit has three sites: the amino acyl binding site, which is shown here on the right side in pink. We also have the peptidyl binding site and the exit site. We're going to go over what these are going to do for us in our next couple slides. But if you want to ever run a reference back to where they are relative to everything else that's occurring, please do so. We then have translation occurring in these diagrams below, and we're going to go through all of this at once. So if anything is confusing, definitely go back and rewatch it. All right, so at first, the mRNA will bind to the small subunit of the ribosome, as we see here on the top left. On the top left here, we have the small subunit binding, and we have the start codon AUG. AUG, as we'll go over at the end, it codes for methionine, which is the start codon. So every protein is always going to begin with AUG. Every mRNA template is going to start with AUG. AUG is going to bring in that methionine right there, and which is the first codon, and that will bind to the A site on the large subunit over here, and then get taken over to the P site. And from there, we're then able to bring in more nucleotides and begin the whole process. All right, so we will now have tRNA strands, which will just be coming in and out. And once they do match the codon of the next sequence, or the next codon of the mRNA, they will bind to this A site here, and that will then push our existing strand, shown here on the left, over to the E site. That's not our original. That will push our original over and to the E site, which will exit, leaving us with our new tRNA connecting to its amino acid, and peptide bonds connecting to all of the others that were there before. And that's what we do to begin. Again, it happens once more. We bring in another tRNA shown here, and we just continue in that cycle until we end. Once we end, we have what is called the release factor that gets pulled in, which is a stop codon of either UAG, UAA, or UGA. Any of those will bring in a release factor, which will kick off our protein, and then will actually break apart the ribosome. It's important to note here that the ribosome does not merely continue on the strand, but does get broken apart and released. And that is translation. So just to end the video, we're going to go over some of the language of genetics, and which is codons. So once again, here we have the codons, which are 3 nucleotide sequences of the mRNA, which code for each of the amino acids. And so we can see here, if we have a UUU or UUU or UUC, that codes for the PHE amino acid. Whereas as you look through these, there's all of the different ones. And here we see that AUG we'd mentioned before. And we also see the stop codons on the top right. Okay, that's that.